Namaste, and extremely sorry for the inconvenience. Am I audible?
Audible, audible.
Audible.
Thank you, sir. So, namaste, and a very good afternoon, one and all present over here. I, Sujit Mandali, Assistant Professor in Chemistry, come convener of this webinar, would like to request our esteemed Chief Guest, Dr. Gopavandhu Behra, sir, to inaugurate the national webinar. Gopavandhu, sir, please.
Good evening to all the participants, the President, Mr. Vice Chancellor, Professor Sanjay Satpathy, the Registrar, the Convener, everybody, good evening. Kalahandi University has gained this way from the pandemic. The pandemic has almost encouraged Kalahandi University to organize several webinars in English, in anthropology, in sociology, in political science, in English, everything. And this is one of the interesting seminar the Kalahandi University webinar the Kalahandi University is organizing. The webinar topic is synthesis design, drug design from a chemist's point of view, not from total point of view, from the chemist point of view. I am happy that they have asked me to inaugurate this seminar. I'm grateful to all of you. I now
Sir, sorry sir, mute ହେଇଯାଇଛି sir. Sir, mute ହେଇଯାଇଛି Professor Gopabandhu sir. Sir, ଟିକେ mute ହେଇଯାଇଛି sir ଆପଣଟା. Sir, ହଁ sir, ଏବେ ଶୁଭିଲା sir. କିଛି ସମୟ ଆପଣଙ୍କର ମାନେ mute ହେଇଯାଇଥିଲା.
ଓହୋ ଯେ ମୁଁ ଯାହା କହିଲି ଶୁଭିଲା କି ନାହିଁ?
Sir, କିଛି ଶୁଭିଲା, ଖାଲି ଏ ଯୋଉ inaugurate କଥାଟା sir ଶୁଭିଲାନି, ସେଟା ଆଉଥରେ କହିଦିଅନ୍ତୁ.
So I am repeating what I said again for 2 minutes. The pandemic has benefited some organizations. Kalahandi University has fully taken advantage of the pandemic and organized several webinars in many departments like English, like anthropology, like sociology, and in other departments. The chemistry department has also not fallen behind, and they have organized a webinar on a very interesting topic like drug design from a chemist point of view. And I'm very happy that I have been asked to be present in this webinar, and I'm grateful to the university for inviting me. With this, I am now inaugurating the seminar on synthetic drug design from a chemist's point of view. Thank you.
Thank you so much, sir. I feel privileged, and it's a matter of immense pleasure for me to extend my warm wishes on behalf of Department of Chemistry, Kalahandi University. I extend my heartiest welcome to one and all present over here for the one-day national webinar on behalf of Department of Chemistry, Kalahandi University.
बाबू रे बढ़िया लगला तोर एटा जो देखिलु मु देखिली आज बहुत बढ़िया लगला सुन हम किछि कहबी केमिस्ट्री रो जो ड्रग्स ऊपर जो कहूछु तापरे मो तो ध्यान धारणा नहीं कौन करी चल बाबू रे एटा पुओ तो पुओ बाबू रे तो पुओ को बाबू कुहा जाए तू कर तो सेटा
हां सर टिके अलाउ करंतु मते सर बढ़िया so I know we all are having some unexpected
proceed on.
Thank you, sir. Thank you. Sorry for the inconvenience. I know. We all are having some unexpected time in this pandemic time to encourage the students and to brush up our knowledge. The idea of this webinar is developed and designed, where our dignitaries will be discussing on drug designing and approach of a chemist. So, at the outset, I extend my heartfelt welcome to the person who has conceived the idea of today's webinar. Yes, the chief patron of today's meeting, our very own Honorable Vice Chancellor Professor Dr. Sanjay Satpathy sir. I'd also like to welcome our beloved Registrar, Sri Padmanochan Bhoi sir. I extend a warm welcome to our esteemed chief guest, Dr. Gopavandhu Behera sir. Thank you sir for accepting our request. I welcome our eminent research persons, Dr. Padmaja Prasad Mishra sir, Dr. T. Senthil Kumar
ଭଲ୍ୟୁମ୍
Armugam
ଦିଅନ୍ତୁ,
sir.
ଭଲ୍ୟୁମ୍ କମି ହଉଛି, ଭଲ୍ୟୁମ୍ ଶୁଭୁନି।
Sir. I extend a warm welcome to our esteemed chief guest, Professor Dr. Gopavandhu Behera sir. Thank you sir for accepting our request. I welcome our eminent resource persons, Dr. Padmaja Mishra sir, Dr. G. Senthil Kumar Armugam sir. Thank you sir for spending your valuable time. A warm welcome to Chairman of PG Council, Kalahandi University, Sri Himanshu Chandra Sadangi sir, Deputy Registrar, Dr. Jayadev Sahu sir, respected members of Kalahandi University, entire chemistry fraternity, respected academicians, my dear students across the state and all corners of the nation. Thank you all for your overwhelming response and gracious presence. I again request you all to be staying tuned with us. Now, I would like to request Ms. Kumkum Priyadarshini, student of BSc final year, to introduce the eminent dignitaries present over here. Ms. Kumkum.
Thank you, sir. Namaskar and a very good afternoon, one and all. I, Kumkum Priyadarshini, feel blessed and honored today to introduce such eminent dignitaries present over here. I am very tiny for this, but I will try my best. I am pleased to introduce our chief patron of this webinar as Honorable Vice Chancellor of Kalahandi University, Professor Dr. Sanjay Satpathy sir. Sir has been guiding us starting from the idea of this webinar. He has served as professor in commerce for over a decade at Ravenshaw University, Cuttack. He holds a vast more than 37 years of teaching experience and served as assistant professor at every corner of Odisha. His contributions to academics is incomparable. He is a magnetic demeanor, histrionic performer, and multidimensional practitioner. He has been conferred with many awards, and the recent one is Golden M Award for Excellence and Leadership in Education. He is the ultimate source of inspiration for all of us. He has also proved being the best administrator, holding many prominent positions like Dean, Examination Controller, NCC and NSS extension programs, evaluated to NAC, and many more. His excellence in every field is tremendous. That is hard to explain in such a short stretch of time. Under his able leadership, the university has started always doing excel from the very beginning. We feel proud to have you here, sir. Thank you, sir. We have with us our Honorable Registrar, Sri Padmalochan Bhoi sir. Sir is a patron of this webinar, a very fine academician. He has his excellence in his subject discipline of Sanskrit. Sir is student helpful and kind by person. He has excellent sagacity in language and literature. His helping and caring nature is the instance of a warm-hearted personality. We all are delighted for your immense presence, sir. Thank you for guiding us and always being humble toward us. Now it's time to introduce our esteemed chief guest of today's event, Professor Dr. Gopavandhu Behera sir. Sir has got his official retirement from his work as Professor of Chemistry from 1978 to 1999 for a record period of 21 years. Besides, his teaching career started from the year 1964 from Ravenshaw College. He holds an incomparable ocean full of experience and patience and passion. To his credit, he has published 120 research papers in various national and international journals. It is important here to mention that Sir has published a review article in Chemical Reviews, that is first time from Odisha, which is widely referred through 300 citations. He has outstanding contributions for students in form of textbook for plus 2 and plus 3 students, which are very popular among the students. He is the person who has popularized the science in Odia and has written a book in Odia entitled as Vigyana o Manavika Mulyabodha. He has produced a number of PhD scholars working across the globe. Sir has visited USA as a Fulbright scholar, a very rare opportunity one gets in lifetime, and also has visited University of Florida and the University of California. He has been bestowed with many awards and accolades, and to name few are V.S.R. Gupta Teacher.
Kumkum, your mic is muted.
Kumkum unmute.
Sir has visited USA as a Fulbright scholar, a very rare opportunity one gets in lifetime, and also has visited University of Florida and the University of California. He has been bestowed with many awards, accolades, and to name few are V.S.R. Gupta Teacher of the Year Award in the year of 1995 by Odisha Chemical Society, and Kalahandi Utsav Shamman in 1999, and Senior Citizen Award by Odisha Vigyan Academy 2011. He is the life member of Indian Chemical Society, Odisha Chemical Society member, Executive Committee of Indian Society of Surface Science and Technology, and was also its Vice President member, Advisory Committee of Regional Science Museum, Pulp and Paper Research Institute, MapCoast of Madhya Pradesh, and Basic Science Group of DSTE. He was a member of the Syndicate of Sambalpur University, member of UGC visiting team. He was WODC member, Appellate Authority for Air Pollution, and member of Program Advisory Committee, Doordarshan, Odisha. The most inspiring thing is Sir has been working relentlessly for the popularization of science and is regularly contributing articles in various newspapers and magazines. After retirement also, he is associated with many social activities. We are pleased to have Dr. Padmaja Prasad Mishra sir with us as our resource person. Now he is serving as Associate Professor in Saha Institute of Nuclear Physics, Kolkata, one of the top institute. He has considerable academic and research experience in his field. He been bestowed with several awards such as Serve Young Investigator Award 2019 and Best Teaching Assistant Award in 2005 from IIT Bombay. His current research interests are gene delivery, gene therapy, and monitoring of mechanisms of complex cellular process, its single molecule solution, and exploration of the activity of DNA repair inhibitors, that is, the future target to improve cancer therapy. We have with us a prominent scientist, Dr. G. Senthil Kumar Arumugam sir. Currently, he is working as a drug discovery scientist at Department of Biotechnology at IIT Madras. His research activities in the field of new drug discovery and development in various sources includes drugs from marine and medicinal plant synthesis, which is developed in IIT Madras, IISc Bangalore, and IIT Kanpur for the past 15 years. He has developed variegated drug candidate in IIT Madras for hypophosphatemia Savarkar 400, that is for kidney disorder, which is marketed as Savarkar, Tolvaptan for heart failure and polycystic kidney disease, hyponatremia for patients which are low sodium level in the blood, and Cystagon for the cystinosis disease, which is for kids with rare genetic disorder. It is important here to mention that recently he discovered drug candidate from originating Terminalia Arjuna plant for inhibiting coronavirus, that is SARS-CoV-2 protease, which is a better candidate for COVID-19 than FDA-approved drugs like Remdesivir and Lopinavir. In fact, his meticulous research provided strong publication record in well-reputed international journals. Thank you for your kind presence, sir. I welcome you all on the board. I have only highlighted here some of the achievements. Please pardon me for that.
Thank you so much, Ms. Kumkum. Now, I would like to humbly request our esteemed registrar, sir, Shri Padmanochan Bhuye, sir, to address the webinar. Padmanochan, sir. Padmanochan, sir, please.
Sachit, you go ahead.
Okay,
Sir
sir,
is
thank you.
probably not connected. You go ahead.
Okay, sir, thank you. Now, I would like to request our esteemed Chief Guest, Dr. Gopavandhu Behra, sir, to grace this occasion. Dr. Gopavandhu Behra, sir, please.
Yes.
Gopavandhu Behra, sir.
Sir,
Gopavandhu Behra, sir. Gopavandhu, sir.
Professor Gopabandhu Behra, sir. Is it audible, sir? Professor Gopabandhu Behra, sir. Professor Gopabandhu Behra, sir, please.
Yes. I am able to. But am I audible to all of you?
Yes, sir. Sir, audible, sir. Audible.
Sir, you are audible. Namaskar, sir. Sir, sorry for the
So-
network problem, sir.
The president of the webinar, the vice chancellor of the university, Professor Satpathy, the registrar of the university, Professor Bhoi, Dr. Bhoi, the deputy registrar, Dr. Jayadev, the convener of the seminar, the distinguished speakers for the webinar, and all the participants, and lastly, the Kumkum, who spoke very well while introducing all of us. Exaggeration sometimes is needed. She has exaggerated a little. Thank you very much, Kumkum. Now I am happy that I have been asked to join this webinar. As I am telling you before, see, drug design is a very wonderful topic. It's a very recent topic also, in one sense. Before drug design, we are able to synthesize millions and millions of molecules, spending crores and crores of rupees, dollars, to develop a drug. Therefore, the drugs, the cost of the drug was becoming very high. Increasingly, people thought that can we now decrease this effort of synthesizing millions of compounds and finally reaching one or two drugs. Therefore, innovation went on. Innovation, as you all know, resonates a sense of purpose in the evolution and existence of human life. The creative capacity of a human mind has always been the source of inspiration. Everyone deserves quality medicine. Now everyone deserves quality medicine, particularly during pandemic type, quality medicine. Apart from the efficiency, or what you call efficacy, affordability and accessibility impact human lives. Now you see, affordability and accessibility. These are two important topics. Therefore, drug design gained importance. I remember when I was in Singapore University in 1970s, perhaps 72 or 73, I don't remember the year, the first paper on drug design perhaps was published, and to my mind, Brufen, a painkiller, was the first drug to have been marketed, which has been synthesized through drug design. Brufen. Then what happens? Why do design a drug? For example, let us say somebody having a pain. You go on trying various drugs and then ultimately reach one. But what exactly now is Once the pain, you understand the signs of pain, which enzymes, which hormone is responsible for the pain. According to the structure, you design a drug. Once you design a drug, then you go and then you try to synthesize. So therefore, our synthesis also became a backward drive. From the molecule, you go on breaking, and then finally saying that these monomers or these particles can go on synthesizing a drug. That is called retrosynthesis. So retrosynthesis helped in drug design, and many drugs have been developed in the world. This is a very, so therefore, in 1970s, perhaps 73 or 74, we introduced a course on drug design. There was a wonderful book on drug design, which covered about 200 pages, and that was taken as a 50-mark course in drug design. Some of university that gave, and after that, of course, at that time there was a lot of freedom with us to design any course. Gradually, the freedom has decreased, and there is no freedom now. So therefore, I congratulate the Kalahandi University. I congratulate the convener to have thought of such a very nice and up-to-date topic, which now demands time because of the pandemic. Now it is necessary to design a good drug for the benefit of the people with less money and more accessibility. I am grateful to you again for inviting me. I am thankful to you. I cannot talk for a long time. Now, I am also thankful to the Vice Chancellor for having asked me to join here. Thank you, Mr. Vice Chancellor, Mr. Registrar, and all of you. Thank you very much.
Thank you.
Thank you so much, sir. Thank you so much, sir. Thank you for your inspiration. Thank you. Thanks a lot for your motivation. Thanks for sharing your experience in this field. Now, I would like to mention to all the participants that the feedback link will be shared by the end of the session in chat section. Filling this is a request for obtaining certificate. E-certificate will be issued within 5 days of the webinar to the registered participants who remain present for the entire webinar session and submit the feedback successfully. So before beginning the technical session, I'd like to request the question that may arise during the webinar should be typed in the chat box so that it will be taken effectively during the question answer session. So without further delay, I would like to request our first resource person, Dr. Padmanabh Prasad Mishra, who will be delivering his talk on drug discovery for SARS-CoV-2 and approach of a chemist. It's over to you, sir. Dr. Padmanabh sir, please.
Thank you, Dr. Mandli. I'm audible.
Yes, sir. You are clearly audible.
Thank you, Professor Behera, for the wonderful introduction about the drug discovery process in a brief, but it was a very wonderful introduction which would actually help the audience to understand the drug development and discovery process in a lucid manner. Thank you, Kumkum, for a wonderful introduction. It's really good. Respected Vice Principal, Professor Satpathy, the Registrar Sir, respected Chief Guest of today's webinar, Behera Sir, all the dignitaries, all the chairs of Kalahandi University, the organizers, my colleagues, my friends, and all the audience. I truly thank the organizers for arranging such a nice conference, nice webinar on this topic, because this is the demand of the situation. As we all know, I'm just sharing my slide. As you all know, that the entire world is going through a pandemic, which just arose because a virus made its first appearance in the animal market of China and eventually spread it all over the world. And that virus, because of that, like infections, hospitalization, and death, it has incorporated lot of fears like this in everyone's life. Every time our day probably nowadays starts with counting certain numbers and they ends by counting certain numbers, whether it's number of infections per day, number of deaths per day, or probably our oxygen level number, our infection level number, or so many numbers every day. So every day we keep checking lot of numbers. Problem is, like, if we check the social media like WhatsApp, we get two completely polarized informations if we check social media like WhatsApp or the news channel, the real TV news channels. In WhatsApp, if we keep reading, it appears like COVID is nothing. You do this, you just take some stimulation, you just take some lemon water, you exercise for some time, and it's nothing. If you check the TV news channel, it says completely different story. Lot of deaths every day. Hospital deaths are too much. So where exactly the story is? Probably somewhere in between. It's not as easy as we read in WhatsApp, or it's not as complicated as it shows in the TV channels, like only dates and dates and dates and nothing else. Somewhere in between. But the fact is, still we are afraid of. That is because there is no targeted treatment. There is no clear-cut treatment that is being defined by any professional body yet. So today, mostly my talk, I'll focus on the drug discovery strategies for this COVID-19, not the drug development, as Professor Behera said, like the retrosynthesis and all these things. I'll not talk about the development process. Rather, I'll mostly concentrate being a biophysicist and working on a biophysicist lab. I'll mostly talk about the drug discovery strategies. What are the strategies that the chemistry community or the scientific community they should adopt, or they should look, or they have been looking into for development of a clear-cut drug or a targeted drug for this pandemic, for this virus. I'll also tell why, like, see, if this virus has made its first appearance in 2019 December, it's almost 1.5 months have passed. I'll also discuss why it's taking time to develop a drug. And if there is no drug, what are the other approaches that the scientific community or the medical community have been following for the treatment of this pandemic situation? Well, as I said, this virus came from an animal and been infecting the humans. This is not the first time that we got a zoonosis disease like this. We had been many diseases like this previously, like there are infections from dog, there are infections from cow which has come to human, there are infections, we keep getting dengue, malaria from mosquitoes, there are cow, chimpanzee, bat. There was a recent disease which was called, if some of you remember, plague. So that came from rat. So that is not, this is not the first zoonosis disease that we are facing. And in fact, this is not the first virus. I'll just change the ledger pointer. This is not the virus which is looking like a corona-like structure, which I'm sure all of us are very familiar with. This is not the first coronavirus that is also detected. There are a lot of corona-like crown kind of structures that have been discovered previously. Like if you check the left side, like NL6, which came in around 1100. Like recent is 2003, which was SARS. In 2012 and 1800 there was MERS, and the recent one is SARS-CoV. Initially, it was thought it is a completely different new coronavirus, so it was called as novel coronavirus. But later it was realized that this coronavirus, this novel coronavirus, has lot of similarities with this SARS and MERS. That's why it has renamed as SARS-CoV-2. And if we check the contagious effect of this SARS-CoV-2, which we familiarly call as coronavirus, just I'll take 1 second. At the timer. Yeah, if we take the coronavirus, then we can see that this COVID-19, which is here, like if you compare influenza, we are just, I'm just talking about one contagious person who has this virus inside. How many persons it can infect? Like influenza can infect 1.5 persons around, whereas this COVID-19 can infect almost 5 people around him. SARS-CoV and MERS, 5 to 8. Measles is more contagious. But the good thing with this SARS and MERS, with whom this COVID-19 virus is very much similar with, both the SARS and MERS are nosocomial. What is nosocomial means? Nosocomial means these infections, the SARS and MERS, mostly happen during a hospital procedure, not mostly outside. During a hospital procedure, this infection was being spreaded. But that is completely contrary with COVID. It's been infecting everywhere. Now it's also proved this COVID-19 also infect through air, which that's why it is called as airborne also. So the infection rate is probably higher than whatever it's been projected so far. Officially it is projected like 4.9, but I personally believe the infection rate is much higher than whatever is been officially projected. How it infects other, it's very known. It's very known. I'm just probably repeating some slides that we must have seen in many media or many newspapers. Like a person with infection, when it coughs, that throws a lot of droplets, water droplets, and that only contains the virus, which eventually enters to the nasal pathway or mouth pathway and get into. It has been seen that when this virus enters to this nasal pathway of this mouth, it stays there for some days because the temperature in this area is comparatively lower than the body temperature. And that suits the virus. The virus stays there for some days before actually gets into the lungs or other parts of the body. Probably that is one reason why the doctors have been prescribing us or advising us, whoever is susceptible for a coronavirus or found to be like infected with coronavirus, to take hot steam, so that, because as I said, the virus in the initial state stays in the nasal airway or in mouth way. So if you take steam, which is very hot, so that will probably kill the virus and destroy the virus, and that is the reason why doctors are advising us to take hot steam. So once it crosses this airway and comes, it eventually ends up having infecting to the lungs, and it by damages the lungs, does lot of infections, and it starts multiplying there. And if one virus has gone inside, it multiplies into millions of virus, and eventually when that person starts coughing, that comes out through the same path that it has entered, either through nose or mouth. Well, so as soon as the pandemic has come, definitely the chemistry community and in general, in particular, or the scientific community in general, they have started inventing into, which is very essential to stop the pandemic, to stop the infection, or to treat the infections. So the entire chemistry community and scientific community, they are from the very beginning, they engage themselves in different aspects of tackling the situation. Like identification and protection of the virus, prevention of the virus, and treatment. And it is good that the virus was being identified in the very initial days, which I'm sure all of us sitting here are familiar with this structure. It looks something like this football with lot of spikes around. So that's why it is this this crown-like structures are there. That's why if you just dissect it, it looks something like this with lot of crowns around, which are called as S protein or spike protein, spike glycoproteins. So the dissection something looks like this. The size of this virus is about 100 nanometer, 80 to 120 nanometer. That means a tip of a hair would contain almost 4,000 to 4,500 virus. It's as small as that. So it's not visible, but it keeps multiplying. So it's a medium-sized virus, but it has a large mRNA, which is the genomic material of this virus. This mRNA, which is a positive charged mRNA, that is enveloped within a cascade, which is mostly a lipid bilayer. Because it's a lipid bilayer, that is another reason why it has been advised to wash anything that is expected to have this virus, to wash with soap, because soap destroys this lipid bilayer that is encapsulating this mRNA genome. So that's the reason soap water destroys this thing. So this is encapsulated with this. There are these spike-like proteins are there. That's why it is called as coronavirus. As I said, these are glycoproteins, like spike proteins. And other than that, it has many other proteins. And this, why I am telling these things, details about the virus is, for treatment of this virus, we need to understand the details of the structural component of the virus. Otherwise, a successful development of a successful drug is a bit difficult. So other than this spike proteins, it has something called this M proteins. If it is visible, the small proteins around here, these are called as the membrane proteins, which are mostly fused on the lipid bilayer membrane. There are N proteins, which are mostly nucleocapsid proteins that regulate the viral RNA synthesis. There are small envelope-like proteins on the bilayer envelope also. These are called as E proteins, and some other structural and non-structural proteins around that. So it's good that the virus is being detected, and it's detected long back, and it is seen that once that virus comes through the human body, it looks for a particular protein in the human body, which is called as a particular, you can tell, enzyme, which is called as SCE2 receptor, where actually this protein, this spike protein, if this is the lipid bilayer structure of the protein, and this is the lipid bilayer structure of the virus, and this is the lipid bilayer structure of the human cell, and this is the spike protein that I just show you in the previous structure. Then this spike protein binds with this small protein that is visible on the cell, which is called as SCE2 receptor. Why this virus mostly affects the lung? Because this SCE2 receptor are more present in the lung cells, mostly on the arteries, the small air holes of the lung. That is the place where this SCE2 receptors are more. That's why this virus directly affects lung. Other than that, this SCE2 receptors are also present on the respiratory airway. There are some SCE2 receptors which are present on the tongue. There are some SCE2 receptors which are present in intestine. That's why some people get stomach upset also. heart, and kidney. And these are the places, because of the presence of this is ACE2 receptor again, this small enzyme, as I am telling it, naming it ACE2 receptor. Because of this presence of this enzyme throughout, this virus mostly attached to those places. And this SARS-CoV is found to be 10 to 20 times bind strongerly to ACE2 receptor compared to other SARS-CoV viruses. How the infection happens? So let's say this is our cell wall. So the virus comes. So these are this Y-shaped structures are this ACE2 receptor that I said. They bind to this spike proteins and they take this spike proteins inside the cell. And once it goes inside the cell, it starts multiplying. I'll in my subsequent slide, I'll tell how this multiplication also happens, which increase the infections and spreads all over the lungs, kidney, and other places. At the same time, whenever there is any infection like this, we know that our body's own immunity system fights, which works like a superhero, which has basically macrophage by help of macrophage, B lymphocytes and T lymphocytes, which are present in the white blood capsule. They keep fighting with the virus or any virus of this kind to protect us, but that is not always sufficient. So that is the reason why either we have to go for a prevention method by taking vaccines, or we have, if get infected, we have to go for a treatment by consuming drugs or by taking prescribed drugs. Almost all are aware about the vaccination process that is going on. I'm not spending time describing the vaccination process. I'll tell how this drug procedure is been going. But though, as I said, though it's like almost 1.5 years, there is no definite drug that has been prescribed by any of the medical bodies. As Professor Behra said, he has said what a drug is probably. A drug, what are drugs basically? If we talk about developmental drug, we should understand what drug is. Any substance having physiological effect for treating, curing, or preventing disease when ingested or otherwise introduced into the body. So any molecule, it could be, it could come from a natural source, it could come from a synthetic laboratory, or if we're talking about natural source, it could come from plants, it could come from fungi, or come from bacteria. So why it's so hard to make antiviral drugs for COVID or other diseases? We should understand that we are talking about antiviral drugs, which is completely different from antibacterial drugs. Because whenever there is a bacterial infection, we can take antibiotic, because antibiotics are specially designed for bacterial infection. And the structure of virus and the structure of bacteria are so different, the nature are so different that antibiotics do not work on virus. That's because bacteria are almost similar to a cell, which has a particular defined cell wall, and bacteria do not need a host cell. But virus, it doesn't have a cell wall. It has a lipid bilayer structure. That's a capsule, that's an envelope, but that's not a cell wall. So all the viruses, they need a host cell, like any host cell. So antibiotics cannot actually go to that host cell, and if it goes, that there are a lot of other complications, like you might have heard about, like multi-drug resistance syndrome, and all these things. Mostly virus also hides into different mechanisms, so that the antibiotic could not even assess the viruses. So antibiotic is not a solution. That's why antibiotic doesn't work. Many questions may arise: if antibiotic doesn't work, why the doctors are being prescribing whenever there is any infections? Why there are like doctors are prescribing to this antibiotic, that antibiotics, and lot, like, because we have been watching the TV and reading newspapers and other social media for a long time. We have been hearing lot of name of lot of antibiotics and lot of TV discussion that is also going on. then why doctors are prescribing antibiotics? No, and that doctors are not prescribing antibiotics to kill this virus. Rather, if there are, whenever this virus attacks to the body, the immunity system suppresses, and because of that, lot of secondary infection starts. Doctors are trying to suppress those secondary infections, which actually happens because of the bacteria. And they are treating that bacteria, the secondary infections. If we take the antibiotics, we are not actually treating the COVID rather. So then why it is taking time? Simply because the drug development process, as Professor Behera was telling, it's not an easy process. It's a very complicated process. Like it starts from targeted discovery, like we have to find a target, then understand the target, design a molecule, and think whether that molecule will bind to that target. Then there are preclinical trials that there are phase 1, phase 2, phase 3, FDA approval, commercial, and if we combine everything, it will take not less than 10 to 12 years. So believe me, if we are thinking of a new drug to come to market for COVID-19, we have to wait not less than 8 to 10 years. That's why waiting for a new drug is not the solution. And if you check the statistics, almost there are 610 drugs which are like under screening, under drug development, 450 trials reviewed by now. 9 are being used for COVID treatment, but there is only one drug which has been approved by FDA. That is because, as I said, it is a complicated process. Well, the good thing is we have identified the virus, we have identified the proteins, like the scientific community, the chemistry community has identified the proteins. So we are almost from here to here. Then it's the job of the computational chemistry to computationally design a drug to see the binding and all these things, and that has been going on, but it will take time. The other approach is, which is called as repurposing a drug. What is repurposing a drug? Because developing a new drug will take enormous amount of time, 8 to 10 years, as I said, that we cannot wait for that. So we have, the scientific community has thought of repurposing of drug. Repurposing means the drugs which are being discovered, which are being used, approved for any other treatments, whether we can use those drugs for this treatment of COVID-19, because we don't have to go for all this preclinical phase 1, phase 2, phase 3, because those are already done for that drug. So those things cannot be, those things are not required. You can probably mute the administration.
Who had entered in? Make it silent,
Sure.
please. All except speaker, all other microphones is to be in mute, please.
Thank you. So,
Sir.
so we are. So the scientific community is, as I said, is trying for repurposing of the drugs, and it has been seen that small molecules, small organic molecules, which have been developed for treating other diseases, has been working very nicely with this, like has been at least helping for treatment of this infection. I'll come into that, but before we see, like my topic was drug development strategies, drug discovery. I'll less talk about drug discovery, but I'll talk about drug development strategies. As I said, for developing a drug, we need to understand the action of the virus, how this virus actually enters the body, what it does, so that we can develop a particular drug for this treatment. So as I said, this, let's say this line that you can see it here is the cell wall, and these are the ACE2 receptors that I was telling you that previously. So our cell wall has ACE2 receptors, and it binds to the spike protein, the S protein of the coronavirus, SARS-CoV-2. So to bind this S protein with the SE2 receptors, it's not like the SE2 receptor directly captures the spike protein and it takes inside the cell. That doesn't work like that. So both the SE2 receptors and the S proteins, they undergo some structural and functional changes before they bind to each other and come inside the cell. The S protein goes a proteolysis, and the SE2 receptor undergoes glycosylation by an enzyme which is called as TM, transmembrane protease, TMPRSS. So these transmembrane proteases are here at the membrane, which does this glycosylation and the proteolysis of these two proteins, and then they bind, and once they bind, they come and come inside the cell. So one of the possible target is to stop the virus right here. So that is the first target that we are going to discuss, to stop the virus right here, binding between SE2 receptor and the S protein. Once it binds, suppose once it binds, it comes inside the cell. It makes something called endosome, and it makes something like this, and gradually comes inside the cell. So once this endosome is formed, the virus stays there. It releases the genetic material, the mRNA to the endosome, and it does so by increasing the acidity of the endosome. The endosomal acidity is something like about 5 to 6, which is more acidic. If somehow we manage to increase the pH of this endosome to more basic, then the virus cannot actually transfer its genetic material and cannot proceed through. Probably that is another reason why the doctors and everyone is advising to consume lot of lime water, the lemon water, because lemon water is more basic, which increases the pH of the cell. And if we take lot and lot lemon water or vitamin C, that helps reducing the endosomal pH. So another target is to reduce the endosomal pH. So anything that can help in reducing the endosomal pH would be a target for a successful drug development. Once the mRNA is released to the endosome, then the mRNA comes, transcripts, and that is done by a protein or an enzyme which is called as RDPP, which is called as RNA-dependent RNA polymerase. RNA-dependent RNA polymerase copies this mRNA, the RNA that has been released from this virus, which is the genetic material, and send it for the next process. So the next target is if we can control the RDPP activity. We stop formation of RDPP, or if RDPP is RNA-dependent RNA polymerase is already formed, if we can inactivate this enzyme, then that can also help stopping spreading of the virus. Because so far the virus has not been multiplied, so we can stop the virus from here. Once the virus process this, then the translation happens and it enters to the nucleus of the cell. This is the nucleus of the cell, and once it enters the nucleus of the cell, it stops the function of the nucleus. So it stops the function of the nucleus by hijacking the nucleus mechanism. Whatever the original, suppose it enters to the kidney cell, it enters to any cell of human cell, so it will stop the original function of that particular cell. That particular cell will stop making any protein that it is supposed to make. It stops the division. It stops division with making the original cell, because this protein hijacks. Because this protein probably can use that ID. Yes. So So the RNA that hijacks the cellular mechanism, so the cell stops making its own protein, which is supposed to make. Rather, it starts making whatever this virus asked him to make. And this virus instructs the cell, instructs the nucleus to make its own protein instead of making what the nucleus is supposed to make. At the same time, this virus at this place asks the nucleus to send any signal of the infections. So the nucleus could not send any signal of the infection. So none of the other organs who are supposed to get the information from the nucleus that the infection has happened would be done, and that's why the infection starts happening. And this happens, and this nucleus, this mRNA enters to this nucleus by help of a protein which is called as impoprotein. And the next target is, because impoprotein actually helps the nucleus, helps the mRNA to go into the nucleus. The next target is if we can somehow make impoprotein inactive. If we make the impoprotein inactive, then the mRNA cannot enter into the nucleus and we can stop the infections. But if nothing works, then the mRNA goes inside the cell, starts making all these structural and non-structural proteins, make the viral capsid, and finally makes the virus and comes out from the cell. And this virus starts infecting the other nearby cells. But whenever this kind of situations arise, as I said, the cell has its own mechanism to stop it. So the body starts releasing something which is called as cytokine to stop this kind of infections. But it is seen that whenever there is a COVID infection, body starts releasing lot of cytokine, which works in which has an adverse effect, and this which is called a cytokine storm, and this cytokine storm infects the lung, and that's why lungs get infected. So another target is if we can somehow stop this cytokine storm. If we can somehow stop this cytokine storm, then we can also stop the infections. So you can see it now, like there are 5 targets that can be, there are other targets too, but mostly people are targeting to this 5 places to develop, to repurpose an existing drug. So now I'll tell what are the drugs that has been repurposed for different targets. Let's first talk about the virus entry inhibitor, which is the first target that I have shown you, where the virus entry of the virus can be stopped, can be inhibited. That is this structure by stopping the acylation or the proteolysis of the S protein or the ACE2 receptor. So it is found that there are 3 drugs. I'm sure again many of us are familiar with this compounds: chloroquine or hydroxychloroquine, imifenovir, or another name is Arbidol, or Comstat. There is a disclaimer here. This should not be considered as a prescription because I'm just talking or uttering the name of this parental compound as a chemist, which has been used as a drug. So these 2 drugs are so far been practiced in different medical body. Though again, I should tell you, none of these drugs are being approved by FDA, but been practiced in different medicals and different countries. So the first molecule I am talking is like this chloroquine or hydrochloroquine, which is mostly used for malaria treatment, which works like a entry inhibitor. It stops the acylation. It stops both actually. It stops the acylation of the ACE2 receptor. It stops the acylation of the spike protein that is around the virus, and it stops the glycosylation of the ACE2 receptor. It inhibits proteolysis of the S2. Also, it changes the endosomal pH, which is the target 2. I'll come in my next slide about the change in endosomal pH. As I said, that is the region. If you remember, I said like once the virus enters to the cell, enters to the human cell, it makes an endosome where the pH is acidic. So this drug changes the endosomal pH and make it more basic. So either chloroquine or hydroxychloroquine, the structure looks something like this. This R, if it is a H, it is called as chloroquine, or if it is a OH, it is called as hydroxychloroquine, and it also reduces the cytokine production. See, if there is a cytokine storm, it is it has having adverse effect. If it is reduces the cytokine also, that has it.
Professor Misra, please unmute. Thank you, sir.
Is it okay now?
Yes, sir.
Yes, sir.
I think I have not missed a lot.
No, sir, it was just for a minute.
Just for a minute, okay. I was talking about this adverse effect of this chloroquine and hydroxychloroquine. So this chloroquine and hydroxychloroquine, though, as I said in the previous slide, it has lot of effect on the target one, like entry inhibitor. It has lot of adverse effect also. Like it has lot of it induces QT prolongation. So any person having like respiratory disease and that has an adverse effect, and particularly people, it should not be combined with drugs like azithromycin and all these things, because people taking azithromycin for any reason should not consume these things. So that's the reason this kind of drugs should be always being practiced under the supervision of a doctor. It induces hypoglycemia. So any person having diabetes should not be administered this drug. It also induces neuropsychiatric symptoms and retinopathy. Anyone having like dual visual syndrome and all these things, this should be strictly prohibited consuming this drug. The second molecule that is like still, I should tell one more thing. In the initial days of COVID-19, if you remember, India was the country who was producing this chloroquine and hydroxychloroquine and sending to a lot of other countries, because people thought we can actually control this virus by administering this drug, because that targets the target one by virus entry inhibitor. And a lot of hospitals in India and in other countries are still using this molecule as an entry inhibitor. Another molecule which has been used is imiperovir. Imiperovir is another virus entry inhibitor. It inhibits the endocytosis, and it is approved in Russia and China for influenza, but it has adverse effect also, gastroenterological upset, and increase the transaminase. So another molecule is Chemist or Nathamostat. The structure looks something like this. It inhibits the TMPRSS. If you remember, I said the enzyme which is bound to the surface, which is called transmembrane transportase, that does the acylation and glycosylation and proteolysis of the proteins. So it inhibits that protein. So this drug is approved in Japan for pancreatitis. It has also adverse effect like GU upset and increase in transaminase. Let's come to the next target. Next target, as I said, next is once the endosome is formed, we can somehow make the endosomal pH more basic. So the drug hydroxychloroquine, which we have seen that it also works as a inhibitor, transmembrane inhibitor, that also works like a modulator of the endosomal pH. It inhibits TMPRSS. and it reduces the pH to alkaline and inhibit the fusion. Then the next target is target 3. As I said, the RNA-dependent RNA polymerase, which stops the transcription, and stops forming the messenger RNA from the genomic RNA of the virus. That is called as RDPP inhibitor. There are 3 drugs that has been practiced or advised by doctors: remdesivir or galidesivir, favipiravir or ribavirin, which stops the virus proceeds from here, from this place. So if we talk about remdesivir or galidesivir, it is mostly a adenosine analog, mostly a nucleotide analog. It works as a prodrug, was tried for Ebola virus, and it's mostly given by IV infusion, and it has many adverse effects like reversible increase in transmittance and kidney injury. So people with kidney injury should not be given this drug. Another drug that is being used for the same reason is favipiravir. We must have heard about favipiravir and remdesivir, and there are a lot of controversial talk, like a lot of discussions, and everything is going on around. This favipiravir is a purine analog. You can see such a small structure, small molecule. As I said, the small molecules are found to be more effective on this virus so far. So it's a purine analogous, and it's used in Japan for treatment of influenza. The problem with this molecule, this drug, is it requires dose adjustment, which is a very critical step, and it has very many adverse effects, like it induces diarrhea, hyperuricemia, and increases transit inventories. Ribavirin, another drug, another RDPP inhibitor, which is a guanine analog, but it requires in very high dose, like and because it requires very high dose, it has many adverse effects like hemolytic anemia, liver damage, and many other effects like that. We can talk about the next target, which is stopping the RNA to enter to the nucleus, which has been done by this impoprotein. Because once this RNA enters to the nucleus, as I said, it hijacks the cellular mechanism. It stops sending signal that the infection has happened. So impoprotein is that protein which takes the molecule, takes the mRNA from cytoplasm to the nucleus, so you can target the impoprotein. And one of the drug that has been used in India widely nowadays is ivermectin, which actually is developed for treatment of antihelmintic and scabies. This is probably the only drug that has been prescribed by doctors that can be taken orally for scabies. But nowadays, doctors are prescribing to this COVID-infected patients to take ivermectin, because this stops the RNA, the mRNA particularly, to enter to the nucleus. But it also has adverse effect like ataxia and drowsiness. And the next is, as I said, once the body gets infected, somehow if the virus enters to the body's own immune response starts, and it starts producing something called cytokine. I'll not go into details, but as I said, particularly it is seen that in this COVID infection, SARS-CoV-2 infection, the body produces unusual amount of cytokine, which is called as cytokine storm. And because the body produces unusual amount of cytokine, it infects the lungs, and the patient who got infected with cytokine gets really weak and reduces a lot of energy. It's basically a corticosteroid. It targets the IL-6 because the body releases many cytokine, like as I have said here. This molecule, this works as a IL-6 antagonist. There are like sarilumab, tocilizumab, and all these things. And it works like anti-VEGF molecule like besanizumab. And another treatment that India was practicing in the initial days of COVID infection, if you remember, convalescent plasma, transference of convalescent plasma, someone who is infected with corona. People used to take the doctors used to take the plasma because that plasma is believed to have more antibody. So they take that plasma and used to put into an infected person. But that is now reduced because that is not even so far approved by WHO, and the effect is not found to be that effective. There is a recent talk all over the media about the Defense and Research Organization's new development, which is called as 2-DG, deoxy-D-glucose, which works in a different mechanism than whatever I said, like, because the host cell that requires a lot of glucose for doing any of its cellular activity. So DRDO, this drug is also not developed recently. That was developed long back, but the DRDO's Institute of Nuclear Medicine and Allied Science, they found that this drug can be implemented here for the treatment because the cell can think this is a analogous of glucose. So so this is a glucose analogous of glucose. So the cell will think that it is a glucose, so it will consume. And if it is really glucose, it would produce ATP and other energy. But if it is 2-DG, it will not produce energy. It will not produce ATP. So the cell will stop working for the COVID virus. Because COVID, once the cell is infected with COVID, it needs a lot of energy. So it's a, as I said, it's the mechanism, the drug action is completely different, but it is found that it reduces the O2 requirement, oxygen requirement of the patients. It's found that it reduces the days of hospitalization of the patient also. It decreases mortality, but it has high risk of cardiac arrest for this drug. Apart from that, there are other mechanisms also, like my lab at Saha Institute of Nuclear Physics, where I am working. We are working on a mechanism where we are finding a particular genome of this virus, and we are working on that still to stop the transcription, the activity of this genome by using a hairpin like a nucleotide sequence. So if we stop a hairpin, it will stop the replication, the transcription of this genomic material of this virus, and it will stop the transmission or replication of the virus. So that this work is also still undergoing. We have got some funding from BRNS, and it's still going on. And this is a review of the different kind of small molecules. This is also a small molecule, a small DNA sequence of 13 to 14 nucleotides. And these are different small molecules that has been targeted at different targets, which are being tested, which are being like practiced by different labs, as I said, but nothing is being approved by WHO. And if I come to a summary, I described about the SARS-CoV structure, how it looks like, what are the genomic material, what are the different targets there. I discussed about the drug discovery pathway, what are the different paths that people follow, the chemical community or the development authority are following to develop a discovery drug. I describe about SARS-CoV-2 life cycle, the pathogenesis of SARS-CoV-2, how it get into the cell and how it make exit, so that the targeting a drug would be easier. And based on the life cycle, I said different targets for this therapy. And I discussed about some repurposed drugs that have been practiced by different medical bodies, by different medical authorities for treatment of this infections. This is a real view of this is the view of the front side of our institute where I am working on. I should introduce this to the students at least, because many of the students sitting here might not know that a institute like Saha Institute of Nuclear Physics, which has been funded by Department of Atomic Energy, exists here, who has like PhD programs in chemistry, in biophysics, in physics, in biology, and every discipline of science. So whenever, like mostly the master students, after completing master, they can communicate, they can contact, they can come to have a PhD degree from. And with this, I thank you all for having patience listening me. And I think the question and answer session will be after the second talk, if I am right.
Yes.
I stop.
The question and answer session will be after the second resource person. Thank you so much, sir. Thank you so much, Dr. Padmaja, sir, for the nice and informative deliberation on the topic drug development of SARS-CoV-2. That is the severe acute respiratory syndrome.
Volume.
Severe. Hello.
You are not heard properly.
Sorry, sir. Thank you so much, sir, for the very nice and informative deliberation on the topic drug development of SARS-CoV-2, that is severe acute respiratory syndrome coronavirus. You have nicely and effectively explained in such a small stretch about drug development, newly developed drugs, and their uses depending upon different types of patients regarding the drug administration. You described about that would really be helpful for the chemist community, research scholars, and everyone else. Sorry for the inconvenience caused during your presentation, sir. Extremely sorry for that, sir. So our next resource person. Now, I would like to request our next resource person, Dr. V. Senthil Kumar Arumugam, sir, who will be delivering his talk on drug discovery and development. It's over to you, sir. Dr. Senthil, sir, please. Dr. Senthil, sir?
Yes. Hello?
Dr. Arumugam, your voice is not like loud. It's not coming clearly.
Hello, sir.
Yeah, it's better.
Yes, sir.
Hello, sir. Now, okay,
Yes,
voice
it is
clear?
audible. Yes, sir,
Much
it is
better.
audible.
Okay. Shall I start?
Yes, sir. Senthil, sir, you can start now.
Shall I start now?
Yes, sir, you are alive.
Okay.
Yes, sir.
So good evening to everybody. So I'm going to present my topic, drug discovery and development, particularly drugs from the sea, medicinal plant, and synthetic small molecule. So the three concepts: one is a natural product synthesis from small molecule to drug molecule. Second is plant natural product. Third is microbial natural product. That is the three concepts we are working. So first topic is drug from the sea. So recently, zingonitide, a peptide originally discovered in a tropical cone. The tropical cone originally discovered from the peptide molecule from this tropical cone snake. The first marine derived compound approved in the USA for treatment of pain. So first marine drug. This one. Next is sea spot trabectin. Another drug they isolated the approved FDA approved as a marine drug for treatment of anti-cancer. So one is this drug approved as a anti-cancer drug in the European Union. Another is zingonitide, another marine derived drug approved as a treatment of pain. Both drug approved isolated from marine. So another marine sponge, they isolated exceptionally potent giant natural product toxin. They isolated from the marine. They approved as a drug. They published in Science. So another Theonilla schweinkoi. So the different kind of polythionamide bioactive natural product. They isolated from the Theonilla schweinkoi. They published in the Nature. It's another drug from marine. So recently, everybody knows COVID-19. The polydipine, a molecule from the sea squid, could be a good COVID-19 antiviral drug. It is actually researchers find the drug 27.5 times more potent than remdesivir against COVID-19. They recently published in the Science journals. That is from marine sea squid. So another edible seaweed, they isolated seaweed extract suppress the superpass the remdesivir at blocking of SARS-CoV-2 in cell models of COVID-19. So sulfated polysaccharide, they isolated from the edible seaweed inhibit the SARS-CoV virus. They published in the Cell Discovery. Both one is from sea squid, another one is edible seaweed. These are all example for a drug from the seashore. So another different kind of drug isolated from the marine. They approved as a FDA approved as a drug. 2004 cardiovascular drug, omega-3 acids, ethyl esters. Another antiviral drug. This one is analgesic drug, zingonitide, anti-cancer. These three anti-cancer drug, they isolated from the marine source. They are FDA approved as a drug. So another topics, natural product, natural drug from the herbal medicine plant. So recently the Chinese anti-parasite drug, they isolated from the Chinese herbal medicine, Chinese herbal garden. They identified the drug for anti-parasite drug. They got the Nobel Prize in 2015, Nobel Prize in medicine for the drug discovered from natural herbal medicine. So another anti-cancer drug from camptothecin from medicinal plant. They isolated from the plant only. Another Plumbago silinica plant. They isolated plumbicin, another anti-cancer drug. These are from natural medicinal plant. So this from literature. 2014 they published natural product drug in clinic. These are drugs are a clinical trial. Anti-inflammatory drug, neurological, cardiovascular, metabolic, immunological, inflammatory, oncology. These are all the drug they isolated. It's under clinical trial from medicinal plants. So if you work in nature, a natural plant or another, you can publish nature very nicely. So these are all important things. Natural product in drug discovery. In advanced and opportunities, they isolated cryomycin in the approved FDA approved as a drug. These are all some important drug they approved as a drug for different disease. They isolate from natural product. These are all some of the example for some of the drugs from natural. These are morphine, analgesic drug, levostine. These are digitoxin. These are another antibiotic. Penicillin for antibiotics, microorganism. These are all flowers from anti-tumor agent. Another gentamicin antibiotic from microorganism. These are another antimalarial drug from artemisinin from herbal medicine, Chinese herbal medicine. Another atopine, another drug for anti-parasite drug. These are all example for drug from natural source as well as marine. So these are all new natural product for a new approved drug for different disease from literature. They published it in a European Journal of Medicinal Chemistry for review article. You can find a lot of drugs from natural. So they isolated, identified, they approved as a FDA approved drugs. So what are the methodology for available for the identification of drugs? but it may be a plant source or from microorganism or sea source, anything. First, we have to extract the extract first, whether it is plant source, bark or leaf or flowers or it may be anything, or from microorganism. We have to extract. We have to dissolve in methanol or ethanol, any extract. First, we have to make extract. Then you have to identify first HPLC, how many compound person in the HPLC pattern. Then we have to identify which fraction is more active, more major. Then we go first, before going to the all the isolation. First, we have to check the activity of the crude extract, whether it is anti-tumor activity, anti-cancer activity, anti-TB activity, anything. We have to first check the activity of the extract. Then we have to check the HPLC. Then HPLC, which is more major compound. For major compound, we can identify by HPLC MS or HPLC NMR, HPLC cycle CD spectra. These three so important technique. We can use the identification of this active compound from the crude extract. You have to use this technique. Finally, we can identify this is a active molecule from the particular extract, which is the active compound. These are all technique need. One is HPLC MS, HPLC NMR, HPLC CD. So another topic, say drug from synthetic molecule, small molecule. Say from andronic acid. This is another. This is a cardiac. This is hyponatremia drug. This drug name is toluene. They synthesize it from small andronic arthamina benzoic acid, andronic acid. They synthesize it from this andronic to this one, another cardiac. This one hyponatremia drug. These two persons, Professor K.C. Nicolau and E.J. Kore, both professor are very top in the synthesis of natural synthetic drug. He is very expert in synthetic drug, synthetic Taxol, vancomycin. Say this one. These are all both professor involved in the synthetic development of this antibiotic and FDA approved by synthetic molecule, synthetic drug. These are all important. Amphotericin B. These are all very important. Vancomycin. So another, so they publish in Nature from this important final molecule, drug molecule. They started from small peptide molecule, peptide to different steps. Finally they achieved in the cyclic peptide. But this is one of the way to from G protein coupled receptor. This drug they started from small molecule. So another way of identifying drugs. This is drug only synthetic route. Synthetic route, we can achieve the drug or maybe plant source or maybe microorganism. So you need different teamwork needed. So one is analytical chemistry, medicinal chemistry, natural chemistry, pharmacology, toxicology. These are all these are major area the people expert need to natural product based drug discovery. So another not only these four people, we need a microbiology expert, microbiology, genetic sequence. Microbiology needed for activity studies, biochemistry people, organic chemistry, analytical chemistry, ecology, pharmacology and medicine people, genomics and bioinformatics people and molecular biology. So different disciplinary people need to discover the single drug. We need different kind of different area, different expertise need to achieve this drug molecule. So what are the process, drug development process? One is first steps is target identity selection. Second is lead discovery. Third is molecular selection for active things. The next one is in vitro studies. We have to identify which is active person by in vitro studies. Next is preclinical. Before going to clinical study, we have to do preclinical studies. Finally, is clinical studies. These are all simple things, but it will take 15 years of time to reach the from here to here. It will take 15 years time to achieve this clinical for a single drug. For it to come to the market, it will take a minimum 15 years, 12 to 15 years need. So it's the same target. First is target selection by using these four experts: bioinformatics, proteomics, genomics, cellular and genetic target. This different group of people need to identify the target selection. Next is lead discovery, need synthetic chemistry, medicinal chemistry, high screening. These are all asset development. These are all need different kind of people to identify lead discovery. Next is medicinal chemistry to do library development, source studies, in silico screening, chemical synthesis. This type of people needed to identify this medicinal thing. Next one is in vitro studies, drug affinity, selectivity, cell disease models, lab candidate refinement. Next is in vitro studies. Finally, is clinical trial and therapeutics. So overall process, drug discovery development timeline. The people will do first drug discovery they will start from 10,000 compound. From 10,000 compound, after bioinformatics, different target selection. Finally, they identified from preclinical study to 250 compound. So from 250 compound, these two processes we need 6.5 years need to identify this 250 compound for 10,000 compound. From 250 compound from clinical study 7 years needed for 5 compound. From 250 to 5 compound. 5 compound to again one drug approved FDA approved drug, it will get another 1.5 years. So overall needed 12 to 15 years needed to one single drug come into the market. So our concept is natural product drug from synthetic small molecule. Second is medicinal plant. Third is microbial natural product. So we are focusing on drug discovery development in the heart disease, cancer, blood cancer, kidney disease, breast cancer, HIV, cystinosis, and tuberculosis. So these are all molecules we isolated, we identified, we filed the patent. In the anti-TB, anti-HIV molecule, we discovered anti-TB, anti-HIV molecule. This molecule under the preclinical trial. This another molecule for SARS-CoV-2 inhibitor. This is for cystinosis, kids with a rare genetic disorder. This is for kidney disease. This is for this drug for anti-cancer as well as blood cancer. This is another drug for anti-cancer drug. This is for hyponatremia, conjunctive heart disease failure. To prevent the cardiac arrest, the people use this drug. We all the drug we file the patent. So everybody knows in India, South Tamil Nadu, Rameswaram, Yelangai, near between Rameswaram, say bridge is there. From Rameswaram bridge, people we collected soil sample from Periyar University. Professor Bala Gurunathan and the team they collected soil sample from the Rameswaram session, Rameswaram temple. The soil sample from that they identified this Streptomyces species. So from the bacteriology from NART, National Institute of Research in Tuberculosis, from this they identified this Streptomyces species. Then they checked with all the bacterial strain, the TB strain. So our part is discovery of this molecule by different technique. So from our side is chemistry. So we identified, we elucidated the structure of this molecule. So identified as a new molecule, new TB and HIV molecule. So this initially we involved in the collection of the soil sample or marine sample from Periyar University, Professor Balagunathan. So in National Research in Tuberculosis, the people bacteriology people they identified this active from crude extract. They identified this the extract active again TB and HIV. So funded by Indian ICMR, funded by ICMR. So IIT. So me and my professor guest involved in this project. Finally identified different technique. This is a molecule drug for anti-TB and HIV. After we are in the 12th year. If so this initially we got from Streptomyces species initially like that. So the microbiology people they extracted active pigment from that extract. This is a crude extract. After this is a TLC of that crude extract. This one top one R1 named as R1. This is R2. This is R3. We separated by using column chromatography. So this after column chromatography separation, this is R1. The top spot is here is R1. Then corresponding isolate after this is a pure compound. So another second one is R2. This is a pure compound after column chromatography. This third R3 is this third spot is R3. This is a final corresponding compound. So this is a crude HPLC of the that extract. This major compound we identified. First, this one is named as a transilmycin. We named as a new name as a transilmycin because a new compound we named as a natural compound is a transilmycin A. Second is a transilmycin B. Third is transilmycin C. From the HPLC, the crude extract, the corresponding the TLC, the RF value 0.8 returns the time in HPLC 4.7, 0.6 returns the time 4.1, 0.3 returns 1.5. 1.5. This after separation of this three compound, mix it together. We run the HPLC. We identified the pure compound. We mix it together, all the three compound, to whether we have written we exactly corrected or not. So the first compound transmission is 4.7. Here also 4.9. Second is 4.1. This transmission will be 4.2. I got it. Third is 1.6. Here also 1.6. So after separation, we mix it together for again rechecking whether it is this three compound again we perfectly separated or not. So finally we separated that we focus on it based on the activity side, that the top spot, the R1 named as transmission A is having more anti-TB activity than compared to R2 than R3. So out of these three spots, R1 having more anti-TB activity than R2. So we focus on only R1. So R1 after identified from HPLC, the purity of the compound more than 98%, more than 90 after separation is more than 98%. This is a UV of the active compound. The UV of the compound is 442. This corresponds to the phenoxysin chromophore present in the molecule. That's why the compound having a dark red color. Chromophore present. That's why is more visible side 442 nanometer. So this is IR spectrum of the compound. The molecule having NH at 10 peptide. This molecule like a butterfly shape. Butterfly alpha ring and beta ring center bond. This looks like a butterfly shape. So alpha ring, beta ring, both they having 10 peptide. Here also 5 peptide things. Another one is 5 peptide. This one is a chromophore. So this both peptide ring attached in the chromophore. So this having 10 peptide, correspondingly we can identify with the IR spectrum. This is amide, secondary amide, NH band and NH. So strongly support this molecule having peptide things. These are the NMR. So these transmissions A and B, C having, see for example, transmission A having only variation only in the proline this position, only keto group. What is our transmission B? Here nothing is there. Only hydrogen without ketone. The third is with OH group. This is a transmission. See, these three molecules same structure basically, but variation only in the beta ring in the proline unit. Here is keto group. Here is without keto. Here is OH group. We named as with ketone. First top part is named as transmission A. Second is without ketone. They are named as transmission B. Third is OH. That is named as transmission C. For example, it will reflect the NMR spectrum. The proline unit, this keto, here is without keto, this is a OH. It will reflect the NMR spectrum, different splitting pattern. So these three compound having same everything, variation only in the proline unit. We can easily identify for NMR different pattern, splitting pattern, only in the proline unit. So these are all molecular weight of the transmission A, B, C, D. So 1269 for molecular weight of A, drug molecule A. Molecule B is 1255. Molecule C is 1271. So we identify these three molecules better, very good anti-TB and anti-HIV molecule, strong anti-TB and HIV molecule. We filed the patent. This molecule dedicated to our India. We filed the patent that the compound transmission is effective against bacterial and viral pathogens. Both the molecule having both character, bacterial as well as viral pathogens. So so most of the people, they will do isolation and identification just to see, simply publish the paper, that's all. But our motto, not like publishing paper, our motto is from lab to clinical trial. Finally, this molecule, the transmission A molecule, is now in the clinical trial. Bioprocess development and preclinical evaluation of novel anti-TB antibiotic transmission isolated from the marine surfaces, now in the clinical trial stage. So now from lab to clinical trial, now we are in 12th year. So 2008 we started, now in 2021. So we've crossed more than 12 years. From achieve this clinical trial step, we need different type of, more than 50 members involved in this project. From bioinformatics, clinical biology, clinical expert, pharmacology people, toxicology people, bacteriology, analytical chemistry, organic chemistry, then again microbiology, biochemistry people, different group of people involved in this project, more than 50 members involved, more than 10 institutes involved, achieve this drug from lab to clinical trial. For safe side, this is a drug molecule. We immediately press meeting. We given the transformation prove effective all type of add HIV for safe side. So for our safe side, new molecule TB and HIV. Scientists come up with a new molecule that is came in the newspaper. Powerful anti-TB drugs in 10 years. Researcher discovered new drug for TB and HIV. It came in the UK BBC news also. These are all new antibiotic found the Rameswaram season. So the already in the there is a newspaper. If anybody interested, you can read the type in Google transenmycin. You can find a lot of information. So another things, prodigiocin, breast cancer. We focus on breast cancer. They published already a lot of people. Prodigiocin is like leukemia. Is a leukemia drug. They already published leukemia, not only leukemia, breast cancer. Prodigy is novel proteins like combined therapy for triple negative breast cancer. They published this drug for breast cancer. The PNAS, these journals are published. But we also identified this. We identified discovered a new prodigiocin. Basic core same, but we got a different modification side chain. We got this also we accidentally got it. The alkaligenous vehicle is from PP11 strain. This after crude extract, we isolated this a new compound and a pure compound. From HPLC is more than 99%. After column separation, this is a pure from based on TLC. This is based on HPLC pure compound. Then finally, this is a UV spectrum of that isolated compound. Isolated prodigiocin. This is the NMR of the compound. This is a molded of the spectrum of the compound. So finally, we identified this is a prodigiocin basic core same, A, B, C, the core same, but we got a substitution here. The second portion we got substitution here. We filed the patent immediately because the very important molecule for breast cancer as well as leukemia, blood cancer. This file immediately filed the patent. This work dedicated to India. So Professor Mukesh Dobli is my professor and my mentor and my both. From the collaboration of University of Kalahandi University and IIT collaboration. So if you want to discover some new drug, we need a collaboration work. A single person cannot achieve the drug for a new drug. We need different type of people, different various collaboration. Then only we can end up with a new drug and new things. So see, if that person worked only with this one, nothing finally and nothing end up. So from Kalahandi University professor, they made a collaboration with us. So we identified this is a new molecule. They finally patent. So we need collaboration. Different people, different university, different kind of people need a collaboration. Then only we end up with the finally drug. Otherwise, nothing. Normal paper only will get it. So similar. Arjuna team. So more than 2000 years. More than 2000 years, people are using this Terminalia Arjuna as a natural remedy for hypertension and cardiac disease. But we worked with Professor Satyanar Kumari and me. We discovered and developed. We isolated 4 compounds from the extract. So, what is the benefits of medicinal benefits of Terminalia Arjuna? Antibacterial effect, cardiovascular effect, antiplatelet effect, antioxidant effect, antifungal, anti-inflammatory, anti-cancer, anti-itching. These are all important medicinal properties of the Terminalia Arjuna plant, particularly antivirus properties. But these are all very important medicinal properties of Terminalia Arjuna plant. We extracted from bark extract. We isolated it with different fraction, extracted different fraction, chloroform fraction, ethanol fraction. See, we isolated oleinic acid from the extent fraction, arginolic acid, arginolitine from chloroform fraction, ethanol from arginolitine isolated from ethanol fraction. as usual, we tried all the EPR activity of DBS, various antioxidant properties, these different compound, different extract we isolate as a conventional method. So we from a DBH activity with the ascorbic acid, we identify this extract, the blend extract having strong antioxidant properties from UV study and EPR study. But then we find, as usual, tried antioxidants and catalyst activity with the various extract with the individual compound and a different extract. The different extract having strong inhibitor property against catalyst enzyme. See, for example, catalase ethanol having more inhibitor properties again catalyst active inhibitor properties because this arginine isolated from the ethanol extract having strong inhibitor properties, catalyst inhibitor properties. So again, we isolated this compound. Again, we checked in stored dose dependent studies. If increase the concentration from 10 microliter to 6 microliter, this compound having still more inhibition properties. From the study, we identify this molecule arginine having strong inhibitor properties from our laboratory experiment. From literature, the cyclosaponin, the tetramethyl they identified, it is already published in the antiviral effect of cyclosaponin and human coronavirus is in 2006. They identified this cyclosaponin tetramethyl as again for anti-coronavirus drug. Then we identified this compound isolated from Germany, some plants. But we got isolated this arginine from Terminalia arginal plant. Both are structurally similar molecule. This cyclosaponin and this arginine having both structurally similar and as well as against COVID activity. Finally, these two parameters. One is experimental studies from our laboratory experiment. Second is from literature. We identified this arginine molecule having strong inhibitor properties against coronavirus. Then what is the symptom of coronavirus? COVID virus people get inflammation, cardiac arrest, blood thrombosis, lung viral infection, high blood pressure, all affected. This Terminalia argina bark extract having strong medicinal property against antiviral properties, anti-inflammation properties, anti-cardiac arrest, anti-thrombosis, anti-lung infection, control the blood pressure and immunomodulator. The bark having all the medicinal properties to cure the COVID disease. What are symptoms are having? This plant having all the medicinal property cure the disease. Then we then focus to change the game to from instead of antioxidant activity, we change it to the potential terminal arginine promising phytomedia against COVID-19, DBH activity, catalyst inhibition, molecular docking, we identified whatever isolated compound from the hexane, chloroform, ethanol extract, all the compound having strong inhibitor properties again coronavirus protease, SARS-CoV-2 protease, 3CL, papillary protease, RNA polymerase. We identified arginine having most inhibitor properties than FDA approved drug, remdesivir 7.6 only. Again, RNA polymer, but this arginine having 8.1 strong inhibitor properties. Then correspondingly, FDA approved lobinavir inhibition properties. Again, 3CL and PCL. But we got it still more by strong inhibition property again. SARS-CoV-2 protease from both arginine having strong in COVID inhibitor, protease inhibitor, then FDA approved drug. Then the big focus. This is the overall conclusion of the drug. So Terminalia argina having we isolated 4 compound. Out of 4 compound, arginine having strong inhibitor property again. COVID arginine having strong arginine catalyst inhibitor as well as COVID protease inhibitor. These all the 3 compared to FDA approved drug remdesivir as well as arginine. So arginine having strong inhibitor then remdesivir. So again we proved with the MDS dynamic simulation also. We got very good result. Arginine having strong inhibitor against COVID RNA polymerase. Then FDA approved remdesivir. Similarly, we isolated another 3 compound from Plumbocos anti-cancer drug. So this collaboration between IIT and biochemistry department from Madras University. So my friend from Madras University department, I am from both are roommate. I am from chemistry department and he is from biochemistry. Both are roommate. He don't know how to isolate the compound, but I know how to isolate the compound. but he know how to check the activity and all the studies. But I don't know all the things. But both collaboration we made finally end up with a very interesting molecule, Plumagin and cis violenica and so after a traditional extraction method, finally identified 4 compound from this is a crude extract 4 compound 1, 2, 3 A, B, C, D compound. After isolation column chromatography, this compound A, compound B, compound C, compound D, we isolated and purified by column chromatography. This is the HPLC of the crude as well as isolated compound. This is HPL of HPLC, Plumagin 6.5. This is standard Plumagin. This is isolated Plumagin. Both are having 6.5. Here also 6.5. Crude is 6.5. Here is isolated. There is a R returns at 3.8. Here also 3.8. And crude is after isolation. This is the crude. This compound having 3.1. This is the after isolation 3.1. So I successfully identified 4 compound from the 3 compound from the Plumagosylinic plant. These are all UV and NMR. We identify all the spectral ready of the compound A, multi-top of the compound A. This is a compound B from UV and IR and NMR and mass, all the things. This is a compound C, the UV and IR and NMR and all the things. Finally identified from different tests. This is the important things. After injecting animal study, there's some from gel documentation, they express after injection of this Plumagosylinic. Actually, Plumagosylinic isolated from the plant. After isolation, we injected the animal rat. After we monitored, then finally identify this one protein expressed. Then we isolated the particular protein. After protein digestion, then we recorded multi-top mass spectra. So finally identified from multi-top spectra, this is from database, we identified DNA mismatch repair protein. So our molecule repair the damage DNA damage. Repair the DNA damage. This molecule having strong properties of DNA mismatch repair protein. Against the DNA mismatch repair protein. From the end of the finally nice things. Similarly, we identified and like collaboration with this biology and chemistry people we involved together. Finally, went up with we identified by different anthro derivative from this cassia auriculata flowers. So this still undergoing a process. So this before that we are discuss about drug discovery plant source and medicine plant source and synthetic all the things. Now development the science from already known drug in the market. So but initially some people are so USA some country initially new approved drug. It will take time to 10 years, more than 10 years, 20 years. It will take time to reach to India for a thing. But in life-saving drugs, Supreme Court they are granted permission. We can break the patent. We can develop the technology, and we can modify the cost. Means the cost reduction. This is the renal tablet. See, the people end up with kidney disease, kidney failure. End up with we need dialysis. But for this dialysis, need this tablet. This tablet only available in the market, in the USA market is around 5,000 rupees. 5,000 rupees, but it cannot accessible here. We need for India, poor people, they need dialysis only need solution, no other option, no drug available in the market. Then Supreme Court, because of the patent with the USA, then the Supreme Court granted permission for us. We developed the technology. We reduce the synthetic method from 10 steps to we reduce 5 steps, as well as cost reduction, as well as the negative effect of this drug molecule. If you take this molecule, the people having strong stomach pain finally end up with some negative side effect. We solve all the negative side effect as well as cost reduction. Now it's 500 rupees only. This drug 5,000 rupees, but only available in the US market, not in India. But we developed the technology transfer to the industry. Now in this industry, with the fossil, in the name of fossil, is just 500 rupees. This is 5,000 rupees. For introduction of this drug, we got to file the patent. The patent also granted. This is a success meetup from IIT director, my Professor Sangaraman. the MIA director, we together team developed the technology. We got the patent brought. We granted a patent. The new drug that getting awarded a British patent. This is the doctor. This one. This introduction of this drug in the India, the doctor got national award from the Nirmala Sitharaman. This came in the newspaper. Similarly, another drug, tolobtain. Need the patient with the low level of sodium in the blood. You need the people with the heart failure. This drug not in the India. This is only in the available in the US market. They isolated, identified, and FDA approved drug, but not in in India. Then we we developed the technology and all the things. Finally, transferred the industry. Now we are going to. It will come in the market very soon. This is synthetic step. We struggle a lot from different. We reduce the synthetic steps as well as all the things. Finally, end up with this molecule with the waste. We reduce the cost reduction as well as the negative effect. All the things we develop the technology and transfer to industry. So similarly, another drug, the kids with rare genetic disorder, the parents struggle a lot. They have to go for abroad treatment. The need is 2 to 3 lakhs need for a 6-month treatment. So we developed the technology, transferred the industry. Now in the market, the people are taking this medicine. So the success meeting, press meeting came. These are kids with rare genetic. These are newspaper also came. This IIT Madras developed a drug for rare genetic disorder. These are all the newspaper IIT Madras developed a drug for rare genetic disorder. So this introduction of this, the doctor got awarded for this introduction of this drug. So another thing, in natural dye, some people will get a wheezing problem or a knee pain, some skin disease. So medicinally active. Some Gandhigram University, they identified a different plant source, medicinal plant source. They identified a very valuable medicinal plant. From that, we developed the technology from liquid to powder form. For example, this is available in particular season. We converted into seed, anotto seed. Then we converted dye. Then the technology for bulk scale production. Then finally we got this powder. Then yarn dye. This transfer to the people will got the people who are got the skin disease or wheezing problem. They will use knee pain. This is one medicinal plant coated this yarn. Now medicinal available. It's less cost but more medicinal available. We developed a technology transfer the industry collaboration between the Gandhigram Industry and IIT Madras. This came in the newspaper, all the things. Then this is all the work and the lab to clinical trial in the presence of WHO chief scientist Dr. Soumya Swaminathan. In presence of Soumya Swaminathan only, we achieved from lab to clinical trial of transin drug, and head of bacteriology and head of biotechnology and head of microbiology at a HIV department, director IIT Madras, director of MIA, director Chemist HOD, different need professor of biotech. We need different group of people need to achieve this. Thanks.
Thank you so much, sir. Thank you so much for your nice presentation, Dr. Senthil Kumar sir. You have described about your research experience. You have informed about the various types of research going on during this pandemic time to fight with the so-called situation of virus SARS-CoV-2. So we are waiting for the potential drugs that may have better effect on eradicating the virus that will bring us a normal time for efforts that has been going on for the goodwill of everyone. So thank you so much, sir. Now it's time for the question answer session. And as we can see, there are a lot of questions that has been asked in the chat box for our first research person, Dr. Padmaja sir. And
Thank you, Suchit. Thank you. Thank you very much. Would I?
Yes, sir. Please.
Say something. Say something.
Yes, sir.
That's a good idea.
Sir will speak after the introduction session. Suchit.
Sir,
Register, sir, you will speak. I will not speak today. You will speak after the introduction session, sir.
No, sir.
Yes, sir. Please wait for the introduction session. Then you will speak and we will end up. Thank you, sir.
Okay, sir. Thank you,
Please
sir.
wait.
Thank you. Oh, thank you.
Go for introduction. It's actually I am astonished with Padmaja, Prasad and Arumugam. You are great. Actually, I am paying my homage to you. You are going on. Please answer the questions. I want my students to be ignited by this.
Sir. So
go for the question answers.
Sir. So first question from our assistant professor from Botany, Khushbu Agrawal madam. Query from biologist point of view.
Sachit, I have answered most of the questions that are asked to me. If there are any new question by the students, they can ask, or you can take the questions of the other speaker, Dr. Arjuna.
Okay, sir. A question by Khushbu Agrawal madam, and that is for Dr. Senthil Kumar sir.
Yes.
Just a second, sir. Khushbu madam, can you ask, ma'am?
Yes, sir. Thank you, sir, for the wonderful presentation. Indeed, sir, you have done a great work that is really helpful to the mankind. And in real sense, we are enlightened by your talk, sir. Sir, my query is that why did you choose this Terminalia Arjuna plant for your drug target? And the thing is that, sir, is it due to a lots of reports about that plant in the literature regarding its medicinal property or anything else?
Actually, that is a slide number 57 is a very important things, because COVID symptoms. First of all, COVID is symptom. What are the COVID symptoms? People will get inflammation, cardiac arrest, thrombosis, high blood pressure, and affect the immune system. But this Terminalia Arjuna having all the whatever negative effect of the COVID patient. That's why I have chosen this Terminalia Arjuna plant. Another thing from Terminalia Arjuna, whatever the structure of the isolated compound similar to cyclosaponin. Already they are proved as anti-coronavirus properties. So that's why based on this literature support our experiment, our lab experiment, the compound having strong inhibitor properties. But inhibitor of catalyst enzyme, but that is also negative. But it's the compound having strong inhibitor property against coronavirus, COVID protease. That's why based on our experimental data as well as cyclosaponin and our Terminalia Arjuna plant, the isolated compound having structurally similar molecule, because they already proved cyclosaponin strong viral anti-coronal virus property. That's why I have chosen, as well as the strong medicinal property of this Terminalia Arjuna compared to other plant. So that's why finally we ended with very nice. We got very nice from docking as well as molecular dynamics. We are trying experimental studies also.
Thank you, sir. Sir, my another query is that how the Arjunantine works in terms of its molecular activity. Means what's the molecular mechanism through which it prevents the activity and all?
First, we have to try with docking on the molecular simulation. Then only we. Only
you,
NI. India only NI Pune only that we have the facility, but that's why we struggling a lot for experimental studies. We are trying. We already made collaboration with USA, New York University. Is under process. We are going to do in vitro and studies abroad, international collaboration. We are trying.
Thank you, sir.
Okay.
Thank you.
Thank you, sir. Another question.
Arumugam, I am just giving you one information. In the forest of Kalahandi, I think you might be knowing it. There is a species twin Arjuna category, so that might be more powerful. So if you feel like if the material switch there, that can be supplied to you by my university.
The voice not clear. Voice not clear. Sir. 1 minute.
It's not clear.
Not clear. Sir. 1 minute.
From the Mahabharata age, there was a particular. Himanshu sir.
No, sir, it is audible here. Might be some problem at that end.
So I will send you that information. It is from the Mahabharata age. There is a Jamal Arjuna, a twin Arjuna plant, which is more powerful than Arjuna, as our scriptures say. So you can add your experiments in that regard. I'll send you, Arugam. I'll send you the things. Don't worry.
Okay. But somebody.
Okay. I'll send you. Maybe there are some problems. We are from the beginning today. We have got the networking problems. Sachit, it is already a lot of time. The questions can be sent to them later on. Now you can invite our registrars to speak on this drug, because it is very old. From Charaka Sanghita, it is also there, which is a transformation of Agnivesha. So invite our registrar sir, who could not be connected at that time, to give his address, please.
Okay, sir. Thank you, sir. Esteemed registrar sir, you can. Sorry.
Suchit, Bhoy sir has a problem in networking, so carry on. Rest of the thing.
Okay, sir. So
now go for vote of thanks.
Sir, actually, presidential address, sir, it's remained. Now I'd like to request the motivation behind the webinar, our Honorable Vice Chancellor, Dr. Sanjay Satpathy, sir, for the presidential address.
Listen, there are two things. One is the network problems on which we cannot do anything. It is out of our range. That we cannot overcome. Okay. Second thing is, listening is a very difficult activity. I don't want to put anything. My students learn whatever they have listened from them today. Okay?
Okay, sir.
Let them learn, let them discuss. Again, we may communicate them with the things and get it clarified. I am happy and I am indebted rather my people of this stature who could devote so much time for igniting our students to be in research. Thank you. Proceed.
Thank you so much, sir. Now, I would like to request the co-convener of the webinar, Mrs. Anusaya Sahman, to propose the vote of thanks. Anusaya ma'am, please.
Again, sir. Very good evening to all. Honorable Vice Chancellor Professor Sanjay Satpathy sir, esteemed Registrar Padmalochan Bhule sir, esteemed Chief Guest Professor Gopabandhu Behra sir, revered resource persons Dr. Padmata Mishra sir and Dr. Bishenthil Kumar Arumugam sir, respected Deputy Registrar Dr. Jayadev Sahu sir, PG Council Chairman Himanshu Charan Charangi sir, Convener cum HOD of Chemistry Department, dear colleagues, participants and my learning students. I owe a great deal to our Chief Guest Professor Gopabandhu Behra sir for his inspiring words and explain how drug is prepared from retrosynthesis process. I am really thankful to Dr. Padmaja Prasad Mishra sir for his nice presentation on drug designing and showed a variety of slides based on drug discovery strategies for SARS-CoV-2 and COVID-19 and explained what is nosocomial zoonosis disease from animal to man, and also said COVID-19 is caused by infection with the severe acute respiratory types of drugs like chloroquine and hydroxychloroquine, which are mostly used for malaria, and also said different type of drugs like remdesivir, favipiravir, ribavirin, etc., which are mostly used for COVID-19. I am very much thankful to Dr. Jisenthil Arumugam sir for his presentation on drug discovery and development and shared a slide based on drug discovery from natural sea, plant and animal, and also explained different drugs like morphine which are prepared from Papaver somniferum, which is a natural plant. It is my proud privilege to convey my sincere thanks to our Registrar Padmalochan Bhuye sir, Deputy Registrar Dr. Jaydev Sahu sir, PG Council Chairman Himanshu Sarangi sir for their inspiration and encouragement in organizing such a circulant task. It gives me immense pleasure to offer thanks to our Vice Chancellor Professor Sanjay Satpathy sir, who guided us for his endeavor and unconditional support for this preparation. At the same time, I offer my heartfelt thanks to our HOD Suchit Mandali sir, colleagues, participants and students for taking part in today's webinar and making it a grand success. I convey my special thanks to our data entry operator Vishnu Prasad Chaudhary sir for his technical support and all other persons who have helped and encouraged us. Thank you.
Thank you so much, madam, for the nice words.
So thanks to all. I again acknowledge the thanks which is given to Padmaja Prasad Mishra, Arumugam. My dear, you are doing a very good work. Our best wishes, and I pray also Ma Manikeshwari, the living deity of the area, to help you, to bless you to go ahead. And I am particularly indebted to a great luminary of the country, Professor Gopabandhu Behera sir, who came and gave his blessings to my students. He is a gem of the area, and I wish the chemistry students will take the benefit from his experience, and in particular, you must read his book that is on science and human values. Without human values, there is no meaning. So, I again thank all of you. The drugs, it relieves pain, relieves sufferings, and it is for common well-being. From the beginning of your career, you should imbibe with that philosophy and go ahead. Thank you. Thank you all. Thank you.
Thank you so much, sir. So,
we can end up here.
Okay, sir. So I would like to request.
Thank you, Padmaja. Thank you, Varugan. We will be meeting again. We will definitely meeting.
Yes, sir.
Sir,
you made us. You made us proud.
Thank you. Thank you so much.
Yes,
okay. Thank you. We'll end up here.
Thank you so much, sir.
Thank you so much.
There is another important meeting for the universities there at 5 o'clock. Huh? 5:30. I have to proceed there.
So, thank you. Now I would like to request to the chief patron of the webinar to officially close the webinar. Sir. Thank you, sir. Thank you, everyone. Thank you for your valuable time.